let's start off with a sound check. Can you people in the back row hear me all right? Can you two in the back row hear me all right? Excellent. I just want to go over a few course matters for people. Susan was able to hold a review session last night. If you were not able to be at it or whatever, the handouts for that are available on Icon. Some copies are also available over here. We took a video of it, and that video will be on Icon shortly, meaning probably, by sometime tonight. There is as well on there practice exams, tutors, slideshows, articles. Angie is going to have her session this week today instead of tomorrow, and it'll be at 4:30, room 433, Bowen Science Building. Trying to think if there's something else that's relevant. Oh, I know. Let me just give you a little forward look at what it's like to take a test for this course, and maybe the people who watch the video will pay attention to this also. This is a fairly comfortable number of people for this room. Many of us are not here right now for whatever reasons. People will turn up to take the test, and it will be much fuller. So I want to ask everybody, first of all, to come as early as you reasonably can. Second, go to the middle of the row before you pick a seat. Don't just fill up the sides. And that'll help a lot at making sure that everybody gets a seat. There are several people who are taking a botany course across the river. I think it's like plant development or something. And I've been in contact with Professor Irish about that. She's going to finish her lecture a few minutes early so that that CAM bus is catchable by those students. So we hope that that'll take care of it. If you still have to arrive late, we'll make sure you have enough time to do the test. Do people have any, another major key thing. Please bring your ID with you to the test. This is not to prove that you are who you are. It's to prove that you put the right ID number on your exam. Miscoded ID numbers cause, you wouldn't believe how much trouble for the size of the mistake. And what we've taken to doing is just simply checking the ID against the test to make sure the two match. And it just really has reduced the number of errors we get by a huge amount and makes things faster for everyone in the long run. So please bring your ID card. If you don't have that, please print off something from ISIS or your bill from the university, something that has your student ID number on it, so we can compare that to what you've coded on the answer sheet. Do people have any other questions in general about the test? You don't need calculators. You don't need slide rules. Yes. It will be multiple choice for the main part of the test. You may be a little surprised. The answer sheet has 10 foils per question. So instead of just A, B, C, D, E, you have choices out to about I. And I don't use all those choices for every question, but for some of the questions there may be 4 choices. For some questions there may be 8 choices. So you just go by that. There will be a bonus question. You do not have to take that bonus question to get 100. It will be a written bonus question. If you decide to try the bonus question, follow the instructions and do it inside the box on the exam sheet. If you need more space than that box gives you, we will have exam books here, but you shouldn't write on the exam sheet anywhere except in that little box. It's a good-sized box, actually. It's plenty big enough for the question we're asking. Does that address your questions? Yeah. I used to give all essay tests for this class, but that was when it was 40. And I have the same number of TAs I have now that I had when it was 40, so it's not as practical. Any other questions about the exam? If you don't like that, by the way, get in touch with your state legislators. Seriously. Is there any other questions people have? Okay, well, let's talk science. That's more interesting than tests. So we can close this up. So we were talking about how enzymes work on Monday, and we looked in some detail at the enzyme mechanism of chymotrypsin. Let me get my pointer going here. Okay. And we tried to press home the idea that the contribution of an enzyme is to lower the activation barrier. The fact that in order to get from A to B over here, you have to go over a hill of energy. That's the intermediate. And what an enzyme can do is bring that hill down so it's not as much energy needed to get to the intermediate state. Usually through a different intermediate, but sometimes through the same intermediate, just facilitated in some way. Actually, I think we did do this slide on Monday. Is that correct? Do people want me to review this slide? You can see, you saw how in the chymotrypsin it was holding reactants near each other because it was a pulling apart reaction, but you can easily imagine that something that's joining together would also be doing that. The orientation, we saw how some of the side chains in the active site could make hydrogen bonds to the tetrahedral intermediate. And so those two things by themselves are a contribution, and in a generic sense, I'm not going to say for chymotrypsin, contribute about 1,000-fold of the enhancement. Some enzymes, but not all enzymes, have to do major changes in the enzyme as they go along. The electrostatic contributions, you could see that in the chymotrypsin mechanism because there are places where there's protonation and deprotonation and so on. In the case of chymotrypsin, actually, you don't want it to completely exclude water because chymotrypsin uses water as one of the substrates. But in most cases, excluding water is a good thing. Let's take a look at another enzyme. This is an enzyme called hexokinase, and it has that name because it is a kinase, and generically what a kinase does is it adds a phosphate group to a protein. Or a phosphate group to something. And so, since the name of this enzyme is hexokinase, we can reasonably expect it to add a phosphate group to hexoses, to 6-carbon sugars. And hexokinase is a beautiful example of how there can be conformational changes when the substrate binds. Here we have hexokinase in the open, unliganded shape. And when glucose is bound, it snaps closed on it like a clamshell. Inside that cleft in the open form are going to be residues that are complementary to the shape that's going to fit into it. And since hexoses have some considerable polar character, some of those complementary residues will be polar ones. So this is an exception to the usual rule that hydrophobic things are in the interior of an enzyme. In this closed state, there are polar residues in there because they have to be polar to touch the substrate, which is also polar. You can see, I think, how in the process of going from this open configuration to this closed one, that you're going to exclude water. That in this configuration, there's a complete contact across the substrate, so there is no place for the water to be touching it. And I wouldn't be surprised if you could have a camera this good, almost watch it squeeze the water off as it closed down on it, but I don't know if we have that level of detail. Because the water is taken out, the enzyme does not have to, or the organism doesn't have to worry about the competing reaction, where you would just simply be hydrolyzing your ATP and producing nothing. This is the specifics of what the reaction is going to look like. Here is glucose, 6-carbon, 1, 2, 3, 4, 5, 6 carbon sugar, very polar with its alcohol groups there. ATP is the common donor for a kinase, although not quite the universal one. And so in the end state, it's phosphorylated up here on the carbon that's flapping in the breeze, so to speak. So that's a 6-carbon, glucose 6-phosphate. We can also learn something about enzyme mechanisms and about protein folding by comparing different enzymes that have certain similarities. And so we can talk about the families of proteases. If we look at 3 particular proteases, chymotrypsin, trypsin, and elastase, we're going to see some similarities in that one. These chymotrypsin and trypsin are both digestive enzymes. Elastase is part of our system for fighting invaders, and it's part of the immune system in effect. If we compare the structure of these three enzymes, look at how similar their tertiary structures are. Remember, the tertiary structure is the folding in space of the whole polypeptide. And when you look at the tertiary structures of these, you can see that in many cases, the secondary structures are in almost identical positions, and the overall packing of it is very similar. If, however, we compare the primary sequence on this, the one, amino acid by amino acid sequence in the linear polypeptide, there is a lot of variation, a great deal of variation. And so you can see that this particular tertiary structure can be accomplished by a lot of different combinations of amino acids. There are many different ways to arrive at the same solution. And what is interesting is if you look carefully at it, you are going to see that it has in the catalytic site the conserved aspartate histidine serine that was used in chymotrypsin. And so although the primary structure is different through a lot of this, it has conserved ones that will fold up correctly and bring together those three specific residues. If you look at residues on the surface, a lot of variation, very little consistency from one of them to the other. So a dramatic example of the relative importance of some residues over others. Some parts of the protein are very forgiving and can accommodate maybe half of the 20 amino acids without any change in protein function. Others, like the histidine, are essential to the enzyme mechanism. If you replace the histidine with a glutamate, you wouldn't expect that enzyme to work anymore. The family to which trypsin, chymotrypsin, and elastase belong is one family of proteases. There are a number of families of proteases. This is from a different family, and it's called subtlysin. This is from a bacterium called Bacillus amyloliquefacus. Okay, I can't read the name. It also is present in a bacterium called B. subtilis. And so the subtlysin comes from the subtilis species, not the one they happen to isolate it from. But the overall tertiary structure, just eyeballing this, does not look very similar to the one we saw in the chymotrypsin guys. So here's the chymotrypsin, trypsin, elastase family. Here's the subtlysin. Really not a lot of similarities, except what's interesting is it still has the aspartate, histidine, serine triad, catalytic triad, the exact same catalytic triad. So this is a completely different way of arriving at the same solution. The primary sequence has no similarity. The tertiary structure has no similarity, but the overall solution to the problem was the same, of using aspartate, histidine, and serine for this mixture of acid-base catalysis and covalent catalysis. We call things like that convergent evolution. There are multiple examples of convergent evolution at many different levels of organization. This particular catalytic triad is so good that there are actually 5 different families of proteases that have used the same combination of residues to catalyze proteolysis. So far, we've only talked about activity, the specific action that the enzyme is doing. And we saw how at the catalytic residues, they had particular interactions with the particular atoms that were going to be part of the final products. But those 3 proteases can do different things. They have different substrates. So if we look at chymotrypsin, for instance, it tends to cleave after large hydrophobic residues. Trypsin tends to cleave after lysines and arginines. And elastase tends to cleave after the small hydrophobic amino acids like glycine, alanine, and valine. So they have different specificities. Extremely similar catalytic residues, very different specificities for the substrate. And the reason is that there's a specificity pocket. So near the area where the catalytic residues are, they're showing this as the sessile bond. This is the bond that the catalytic residues are going to cut. That's what sessile means. It's Latin for cut, cuttable. And if you look in chymotrypsin, the contours of the enzyme have a deep pocket that has around it some, here's a couple of moderately hydrophobic residues. So it's a deep and not very polar pocket. So you can understand how that could accommodate large hydrophobic residues. If we contrast that with trypsin, you still have the two glycines there, but now in the bottom of the pocket, you have aspartate 189, and it's going to have a negative charge there. So you can understand how, biggish amino acids like lysine and arginine with their positive charges could fill that pocket and then interact with the negative charge of the aspartate. And then elastase, which is only going to accommodate small hydrophobic amino acids, is sort of filling in the rest of that pocket by sticking in a valine and a threonine where these had just plain glycines. So you can see that the residues in the specificity pocket confer spatial properties on the enzyme that distinguish between the possible substrates. That what residues are sticking into the substrate specificity pocket are going to determine what substrates can interact with that enzyme. And you can see then that a very small change can change the utility of an enzyme quite dramatically. So if you were starting with chymotrypsin, a point mutation here at aspartate would give you an enzyme that had the activity of trypsin. There are probably more changes than that between the two of them. But that difference alone, that a single amino acid would change you from large hydrophobic to positively charged side chain. That's a big difference. And it could be accomplished by a single nucleotide change. Well, maybe not a single nucleotide between serine and aspartate. It might take more than one nucleotide. I don't want to commit myself too deeply there, but a single codon change. Here you need a couple more changes, but not a large number of changes. Chymotrypsin is an interesting example of the ways in which many proteins take steps towards achieving their final structure. And this may be partly because folding up as a precursor may help ensure arriving at a final state, or it may just be that they want to control the activity of the protein. There are a number of reasons why it might happen. But the way chymotrypsin is made is it starts off as a long protein called chymotrypsinogen, and the inogen part means something that can make chymotrypsin. So the inogen ending will occur a number of places. So this is chymotrypsinogen, the protein that can make chymotrypsin. And the other digestive enzyme, trypsin, can attack chymotrypsin as a substrate. And when it does that, it cleaves it at one spot after an arginine, because it's trypsin. And now this is one form of chymotrypsin, meaning it has the activities of chymotrypsin in the sequence of chymotrypsin, but it's bigger than the final product. So they call it pi chymotrypsin, and it is enzymatically active now. It will cut itself, so chymotrypsin, as pi chymotrypsin or other chymotrypsins, can then attack again and cleave off that little piece here of the serine to arginine there, and cleave a second time at the sort of block in there. These are threonine to aspartate. So it's leaving two smaller pieces and one that is now the mature active chymotrypsins. This is alpha chymotrypsin. This was delta chymotrypsin. I suspect these were named for where they ran on gels, that alpha is Greek for 1 and beta is Greek for 2 and so on. So alpha chymotrypsin is probably the fastest running of them, and delta and pi would have been slower because they're bigger behind it. But you can see here how much processing goes on for something as simple as a protease whose only job is to break a peptide bond. Some other things have considerably more processing than that. So why is it that chymotrypsinogen is not active and trypsin is active? Chymotrypsin is active. What happens when that bond is cleaved to activate the proenzyme, as we call it? If we look at the protein structure of chymotrypsinogen, the precursor, we already have the ASP-HIS-SEER sequence that we expect in the mature enzyme. It's not that we don't have the residues there that we need for catalysis. But what's different is that when these other parts are cleaved off, we now have a change in conformation that exposes a specificity pocket. You have a place for the side chain to bind and select things, and the oxyanion hole that could stabilize the tetrahedral intermediate is now in the right position to do that. So this is a good example how activity is not just restricted to the most intimately catalytic parts of the active site. It can also involve other features as well. If we take in this whole thing, the catalytic residues and the specificity pocket and all the stuff that sort of is in that area, that would be the active site. And an active site can encompass the catalytic residues that actually do catalysis. It can involve substrate specificity and so on. Proteases might seem like kind of a boring enzyme if all you think of them doing is digesting steaks, although digesting steaks is a lot of fun. But proteases play a role in a remarkable number of things. And I certainly can't exhaust all of them, but we can talk about one or two of them in some detail to give you a sense of how you can accomplish interestingly different things by small changes in an enzyme and how big the role is of something as simple as a protease. When you have a cut and you start to bleed, at some point in most of us, that cut will stop bleeding. There will be a clot formed. And for that clot to happen, there has to be these fibers made of a protein called fibrin. And you can see it looks kind of like a web here of fibers. That's why it was called fibrin. But we don't clot all the time. I mean, our blood is running around in our body, most of the time it doesn't clot. So how does the body stay balanced so close to being able to clot when you need it and free of clots the rest of the time? And this is going to be very important because controlling of clotting is going to controlling strokes, controlling heart attacks, and things like that. So here is, it's a trick like the trick with the chymotrypsin and chymotrypsinogen. Here we have fibrinogen, so ogen, that ogen ending again. So this is the thing that gives rise to fibrin. And fibrinogen by itself doesn't make structures. But when a protease called thrombin digests it selectively, you then have a fibrin monomer that is able to make polymers. And you can now have it line up side by side and make extensive fibers. And then those fibers trap these little structures, which are called platelets. The platelets have other proteins in them as well, and they're also able to contract. So each of those little sort of starfish-shaped things will at some point contract themselves, and that's the point at which a clot kind of turns hard. The platelets have contracted and squeezed the water out. And platelets are important for clotting too. It's not quite as simple as that. It certainly, what we talked about right now is thrombin and fibrinogen going to fibrin. So this is certainly a true part of it. But that just raises the question, where did the thrombin come from? And what we're going to see in the clotting cascade is an example of how enzymes in sequence can amplify signals. And so you have a series of proteases that are going to target another protease. Thrombin is the one that actually turns fibrinogen into fibrin so you can actually clot. But thrombin itself has a profactor called prothrombin. It gets activated by a protease that has a couple of names because people discovered it along the way. That had a profactor. It, again, is digested. You don't need to memorize the sequence of these proteases, trust me. But what I do want you to take away is that there's a cascade of proteases, each one of which is activating the precursor that's inactive. And since one molecule of thrombin can activate 100 molecules of fibrinogen, and one molecule of XA can activate 100 molecules of prothrombin, what you're getting is 100 times 100, so forth, amplification. So between this one active molecule, you can get millions of fibrin molecules. So by having a cascade of enzymes, you amplify the signal at each step. So that's good. So that means you only need a small signal to get a big result. And in the case of clotting, the way they do that is they make sure that the control of converting prothrombin into thrombin is locally controlled. And so once a little bit of thrombin is activated, that little bit of thrombin can now start the chain that will result in a lot of thrombin and a lot of fibrin. And if you look, here's our old friends, asp, his, seer, the same catalytic triad that we saw in the gut, but here it's in the blood clotting. So reusing things, kind of like a tinkerer who, you know, finds the screws in his workshop and uses them. Another way you can regulate proteases is by having inhibitors. So in other words, you can have the activity turned off until you need to have something happen. And this is an example of trypsin here interacting with pancreatic trypsin inhibitor. Now you can understand perhaps if you think about the anatomy of this, why the pancreas would have trypsin inhibitors. The pancreas is making all the digestive enzymes. You don't want to digest your own pancreas. And so the pancreas, in addition to making the enzymes, makes inhibitors that control them that then are going to be distributed or taken away once they hit the stomach or fall off at the pH and something like that. And so here you have a trypsin inhibitor bound to the trypsin. Some of these inhibitors are quite high affinity. And in this case, what it does is it mimics the substrate, but it can't be cut. The particular structure of it as a polypeptide doesn't let the catalytic triad cut it into two pieces. And since it won't get out of the way, nothing else gets eaten up either. These protease inhibitors play a role in a number of places, and this is one that affects millions of people. It's a good thing sometimes to break down proteins. So for instance, when you've made a clot and you want to clean up the wound site as you're rebuilding the skin, you have to get rid of some of the stuff that's in the clot because that's not the final stuff you need. Proteases can help with that. When you're looking at, in development, for instance, the earliest stages have hands that look kind of like paddles, and part of what happens is there's death and proteolysis that sets the fingers free to become independent digits. This is an example of the role of proteases in the lung. And this is a beautiful light micrograph showing you what a healthy lung looks like. Here's the big, beautiful alveoli, so you can see that you get a lot of air into that, the nice intact passage of the capillaries around it, so the red blood cells, as they course path, will be picking up oxygen and letting go of their protons on the bicarbonate and so forth. So this is the healthy lung. In a disease called emphysema, that architecture breaks down. So here's an example of emphysema. And you can see that if this is an alveolar space, it's really quite collapsed by comparison. The walls of it are not very thick. It's smaller. It's really clearly going to be much less functional than the beautiful architecture of the healthy one. And part of what happens is that in the healthy lung, there is a protein called alpha-1 PI, alpha-1 protease inhibitor. And that alpha-1 protease inhibitor has its job of inhibiting elastase most of the time until the appropriate signals come for it to work. There are some people who do not have that protease inhibitor. And those people are prone to developing emphysema just because they're missing the inhibitor, and that's the bad luck that they have to live with. But there's also a way of hurting that elastase or failing to control that elastase through smoking. So if you look at alpha-1 protease inhibitor in the lung, in those who are smokers, you can see that the methionine side chain, so methionine has 2 carbons, a sulfur, another carbon, so it's this pretty hydrophobic side chain. Methionine is oxidized. Here's a sulfoxide now, and you can see that carbonyl oxygen. You can imagine that's going to be a much more polar, much more hydrophilic residue. And so that ends up damaging alpha-1 protease inhibitor. Without the alpha-1 protease inhibitor, you don't inhibit the elastase. With the elastase active, you end up with your lungs looking like that. And there are some new slides that aren't in your handout, so be patient. This is showing you how this one little bit over here is enough to kill the inhibitor and then end up damaging the lungs. Some of you may have seen that Senator Barack Obama is quitting smoking. I think that's cool. This property of having active residues that are the catalytic residues that play a particular role chemically, sort of in a cage of other parts of the protein that control things like how the substrate binds, is a clue to how the variety of enzymes has come about. And this is an example of that kind of variation. So you can talk about having a scaffold, a region that is fairly constant between several different enzymes with catalytic residues that are preserved between these families. And, oops, dang. Oh, what did I do? Okay. You can then look at the rest of the architecture of these enzymes, and in one family member, here we have family member X, we've got loops that stick up like that, and those loops then favor the binding of one particular substrate. And in a different family member, we have a different set of loops sticking up. And this is shown diagrammatically on the chart of the gene over here, that you have maybe two sites that are the catalytic residues, and then you have these other colored sites that play a role in substrate binding. So gene Y, if you stretch its gene out, would look like that. Protein X has its gene looking like this, where you have different colored thingies. And if you have recombination between this and that gene, you have now a piece of DNA that would look like this. And when you let that fold up, you have a new member of the family that has some similarity to both of them, but because there's some novelty, because they're put together in novel ways, they can have a novel substrate. We see this kind of pattern by looking at the genes of lots of enzymes. So it's pretty clear that a lot of the enzymes that now exist in living cells arose through recombination of previous enzymes. It's been taken a step further by having that kind of evolution actually occur in the laboratory. And so the kind of loop switching I was talking about in nature would look something like this. You'd have your undifferentiated scaffold, the sort of basic scaffold with its catalytic residues, and it might go with one set of loops here to become one particular enzyme, a different set of loops to become a different enzyme. Some biochemists have worked that, in effect, in the laboratory. They've taken one of these enzymes and identified in it what they believed was the core of it, like that. So they took off the stray bits that weren't part of that core, and then mutated it to give it random loops and selected for enzyme activity until they got a bit of the enzyme activity that they wanted, and then they could again mutate and select. And so they can select and reconstruct now an enzyme that has a very different specificity than the one from which they started. So what we've been able to reconstruct, by genetic analysis has been mimicked in the laboratory. And I think that's a very powerful analysis. I'll make sure that these slides are available on the web as well. We've finished a little early. Would people like some review over some of the other material that we've discussed? Any questions in particular? I can refresh, if you like, some of the things we did in the enzyme energetics. I don't know whether these are things that are going to be when we started talking about enzymes, we talked about reaction coordinates. And And this is actually connects back all the way to chapter 1 when he talked about free energy changes. And so if we have a starting state and an end state that have equal free energies, there's no real reason for it to exist in one state versus the other on energetic grounds. So getting from one to the other will have to pass through an intermediate. So this would be the transition state, and this is the free energy of activation. You have to get at least that much energy into the system to arrive at the particular conformation of the transition intermediate. And so it's this activation energy that's going to be one of the key things at looking at what enzymes are doing or not doing. This activation barrier is there whether or not there's an enzyme there. So if you have a reaction happening at all, there is going to be some kind of an activation barrier to that reaction. If we look in the absence of an enzyme at the relationship between the starting material and the products, if the products have a lower free energy level, so it's a negative delta G, a negative delta G will indicate that that reaction can occur spontaneously. It's going downhill, if you like. If, on the other hand, it has a positive free energy change, that there's more free energy in those products than not, something has to be put into the system to get that free energy change to occur. So the change in the free energy between the reactants or the starting material and the products is only an indication of, can the reaction happen spontaneously without the input of energy from something else? This activation barrier, the activation energy, is going to determine the rate of how fast it goes. That just knowing the difference of the reaction overall cannot tell you the rate, because the height of that activation barrier may be different. And this is the killer diagram, if you like, that what the enzyme is doing is simply making it easier to get to that transition state, lowering the activation energy, and now more molecules will get over the hill and run downhill to the products. It was always downhill from reactants to products, or it wouldn't go spontaneously. The difference is there's now less energy needed to get through the transition state. Is that clear to people? Does anybody want me to explain that at all anymore? Are there other questions that we've talked about during the first 5 chapters you'd like me to review or discuss? Yes? Sure. Give me just a second to get that one up, and I'll park this for the moment. It's a good question because it really does come into a lot of things. Here we go. Open. Okay. So, shall I look at our titration curve? Would that be a good place to go? Okay, let's hit a titration curve. Come on. Work. There we go. Sorry? Yeah, sure. Why is this not showing me anything? Micro sloth. Ionization. This is a place to begin. All right. So I'm going to back up from this one. Let's see. Sorry, which one? You mean if I were to close out one of the other PowerPoints? Okay. I'll try closing that out. Okay. So, yeah. Well, this is certainly one of the things we need to think about. If we look here at, here are two extremely common functional groups. This is a carboxylic acid, and this is a primary amine. At pH, at neutral pH, at pH 7, there are going to be equal numbers of protons and hydroxyl groups, equal amounts. The further you go small in pH, 6, 5, 4, 3, 1, down that way, the more protons there are, the fewer hydroxyls. The further you go up towards 14, the more hydroxyls, the fewer protons. So down towards pH 1 is getting increasingly proton rich. Going up to 14 is getting increasingly proton poor. If we look at the carboxylic acid and we start raising the pH, the environment is getting poorer and poorer in protons, and at some point its proton will fall off, and now it's ionized. This is a deprotonated and an ionized group, and it will remain deprotonated and ionized all the way up for the rest of the way up. If we look at the amine group down at that acid pH, it will be protonated. Both of those are protonated. But an amine group that's been protonated becomes positively charged. And as you go further and further into a protein-poor environment, at some point you'll get to a point where it loses that proton and now is deprotonated and is unionized. Ionization refers to the presence of some charge on it. Protonation refers to a reversible loss or gain of protons. And if you're neutral and you gain a proton, you get positively charged. If you're negatively charged and you gain a proton, you become neutral. So as you drop the pH, this ionized carboxylate is protonated and now is unionized. As you drop the pH from neutral, it remains ionized. Is that feeling a little bit clearer for folks? Okay. These two appear in so many places that it's worth being comfy with them. And of course, if we look at the pH of the body, which is pH 7, both of these will be ionized. Both of them will be charged. The carboxylate will have a negative charge. The primary amine will have a positive charge. So the carboxylate will be deprotonated and the amine group will be protonated. Let's see. So here we're looking at the relationship of pKa and pH. Is this another one that you wanted to have looked at? Okay. Something like hydrochloric acid simply falls apart into chloride, ion, and proton, into completely charged pieces. There are a lot of groups that don't go all the way all at once. We call them weak acids or weak bases. That is, they will, some fraction of them will lose their proton and some of them won't lose a proton. And we can look at that property. I'm going to fix my cursor if you don't mind here, because I'm getting crazy on this here. There we go. So if we start, this is a titration curve for acetic acid. So it can undergo that, this would be the protonated form that we saw in the previous slide. This is a deprotonated form. If you're starting at a low pH, so this is starting below pH 3, basically all the molecules in this solution are going to be in the protonated form. pH 2.5, there's lots of protons around, proton-rich environment, fully protonated, unionized, for the case of acetic acid. As you start to raise the pH, there are fewer and fewer and fewer protons around, and bit by bit, that protonated acetic acid is going to lose a proton. First these molecules will, then those will. It's a statistical thing. It's not all at once. And as you get poorer and poorer in protons, getting closer and closer to pH 7, more and more of these molecules will lose their proton. But it doesn't happen as a straight line. If we look at it, it has a curved character to it, sort of a sigmoid on its side character, if you like. And this is an inflection point. This is the point where it's changing from curved up to curved down. And this midpoint is the point at which the number that's protonated is equal to the number that's unprotonated. And the number that's ionized is equal to the number that's unionized. And that would be true whatever groups you're talking about. The midpoint is that point where you have half of one and half of the other. Add protons, you'll protonate the thing. Add protons, you'll protonate the thing. Take away protons, you'll deprotonate the thing. At the midpoint, it's half and half. And then you keep making the environment poorer and poorer in protons, and we have, now we end up, at the end point of it, it'll all be in the deprotonated state, the carboxylate. And that same logic can be applied for the amine group with a different pKa. The name we give that midpoint where half of them are ionized and half of them are not, is pKa. We arrive at that through some math, but in practical terms, the pH at which half are ionized and half are unionized is the pKa. So pKa's tell us something about the behavior of a group in different pH solutions. The acids all have pKa's below 7. The bases have pKa's above 7 in general. We can also take a look, if you like, at some of those specific groups that let's see now. I think there were some examples of, maybe the amino acids were in the amino acid chapter. Do you want more on that, or is that a good handle? Do people want me to look at some of the amino acids and talk about their protonation, deprotonation, or what? What would people have a question about? Yes? Some amino acids, is that what I'm seeing? Okay. I'll probably need to close this out then and open up the proteins. That means we can talk about anything with proteins. Okay. That. Proteins is chapter 4. Here we go. So part of what gives proteins their wonderful diversity of behavior is the fact that with the 20 amino acids, you have really a lot of chemistry to play with. And if it, okay. I've closed the other ones down, and it's still moving slowly. Oh well, it's cold outside. Maybe it's sluggish. Waiting. Okay. There we go. So this would be slide number 10 in chapter 4. I want to get it open. Come on. Okay, there we go. Square. Okay. So here it's just simply represented as the name of the groups. You can see that aspartic and glutamic acid both have acid pHs. It's not too surprising. So they're going to be ionized and deprotonated at pH 7. Glutamic acid is almost, 3/4 of a pH less acid, and there's just the one more carbon in that side chain. So this gives you a little example of how the details of the side chain structure allow the proteins to be tailored very finely. So you can get a difference of almost a pH unit out of just adding a single carbon. Here's histidine. Its pK is surprisingly close to neutral. It's not quite neutral, but it's pretty close to neutral, which means that even at pH 7, you're going to have some protonated and some unprotonated. At pH 7, it's going to be mostly unprotonated, but there'll be some protonated ones there. Now what's interesting is inside of a protein, a side chain can be in a particular environment that changes its pKa. So a histidine inside a protein might have a pKa of 8. Doesn't happen often, but it's easy for it to have a pKa of 6.5. And so the environment of the protein can influence how likely that proton is to come on or off. Cysteine is sort of the partner on the other side of neutrality. So it's close to neutrality, but on the other side. So at neutrality, it's mostly protonated with a few unprotonated. But that fact that these two are fairly close to neutrality means that you see them over and over again at enzyme active sites because they're capable of gaining and losing protons under fairly mild conditions. And so you have histidine in this family of proteases. We've seen them over and over and over again. There's a whole separate family called cysteine proteases, which have a related but not identical mechanism. So histidine and cysteine are common residues to see when you have acid-base catalysis because they can undergo this, associating, dissociating so easily. And then we have our two strongly basic ones, lysine at 10.3 and arginine at 13. So these guys are going to have a positive charge on them at basically any conditions that biologists look at. I don't think there's ever a time, of them being deprotonated. So they are positively charged under essentially any condition that a living system is likely to want it to do. Well, it is 4:20. I can go on doing this for a while if people want, or you're welcome to leave. I mean, you're certainly welcome to leave anyway. But if people want me to continue doing this for a little bit, I can. Sure. Okay.
Hydrophobic effect is the reaction van der Waals
hydrophobic effect is not due. Let me turn this off so we're not recording. Hydrophobic effect is not due to interactions among things that are hydrophobic.
Oh,
It's the fact that water molecules can't interact with the hydrophobic things, and so there's a break in the network. They have to. Yeah, I will be, but I need to put it in.